I'm Paten Agre. I'm a molecular breeder from IT, Ibadan, Nigeria. So what I'm going to present here is just a summary of the different activities we have been doing, and then how we are using a genomic tool to improve our selection and also anyway. So what happened? Oh, okay. I think I got the script now. Okay, so the outline, I will just talk briefly on the marker detection by using the genome-wide association and also QTL mapping in a biparental population, marker validation and deployment in the breeding program, and then a cross prediction, and then just a brief summary. So in the breeding program, we have been using several types of sequencing from whole genome sequencing, WPN, and then we have also some GBS data, including working on ALATA and also Rotunata. And then we are using also diversity array technology, including and also make use of Intertech, mostly for routine activity and so for validation precise. So in the breeding program, we're able now to have kind of like a roadmap for our SNPs calling using a whole genome sequencing and then also GBS. So we have documented the process, and then we have a workflow ready for use. So the first activity we did is genome-wide association study in the breeding panel. So we have kind of like up to 400 accession, whereby it's kind of like material from early generation to advanced population, and then we combine them. And then we make use of a multiple random selection model, and then we're able to identify some genomic region controlling some key traits in the breeding program. This work has been published, so sorry I could not present the full detail, but if you want to know more about the work, how we, the different method we use, you can see, you can have more information. And also we make use of GWAS Poly, which is a tool developed by the Polyploids team. And then we make use of 4 different models: the general, additive, dominance recessive, and also a dominance alternative. And then we're also able to identify some region controlling the flower intensity, plant cells, and then the plant vigor, including some other key traits. The plant cells is very important, is very crucial for us because it's too difficult at the early generation to be able to predict the cells. You have to wait for almost 3 years to be able to have the information about the sex. So we're able to identify a very good marker that we are using in the breeding program and then to predict the sex. We did also the same thing for ALATA. As I mentioned before, we are working on 2 main species, Rotunata and ALATA, and then we use the same method on ALATA. So we identify also genomic region. So you have this one also will be published, so you can have more information. Now, when we start the mapping population, it was not easy for us because the first genetics map we built, I remember how I disturbed Marcelo, was not easy. The first genetics map we built was not really good, a lot of gaps between the different linkage, and also a lot of recombination between one genetics map between one linkage group to another one. And then we made use of the GWAS Poly, map poly, and then we're able now to document the process in each of the analysis, and then we predict also the ploidy level in the breeding program. And then you can see also the quality assessment we use both for the accession and also the marker. So we're able now to remove those markers and then also the genotype that are not respecting our filtering criteria. And then you can see also we got a very nice frequency recombination when we split them across all the 20 genetics map. Before we could order the marker across the linkage was not a good, but at the end we're able now to get a very nice recombination fraction, and then help us to build a very nice genetics map that we are using in the breeding program. So we compare our genetics map with the ALATA reference genome, and then we got almost 95% of a resemblance, which means our recombination in marker order are in very good shape. This work also has been published, and then you can last year we can have more information about the way we proceed about it. And then we are also identified 4 SNPs marker that are associated to the disease, and then we have converted them into a competitive allele SNPs marker that we are using also in the breeding program. So for the marker validation, we are currently working on 7 traits, but I will not be able to present all of them due to the time. So I will only just present on the plant cells, and then you see the way we have our marker accuracy. So what we did here for the validation, working with Intertech, we selected 184 sample, and then with known cells and unknown cells. And then we send the sample to Intertech alongside the flanking sequencing, which was developed with Illinois University. So what we did is just those plants that we know the cells, and then the one that we don't know, we just run a pairwise ordinary linear regression through a bootstrap. So we're able now to predict the correlation. So for plant cells, we got for the marker, for one of the marker up to 91% accuracy, which is very good. And then we have deposited this information into the EIB, so anybody can take the flanking sequencing, use it. Now, let's move to the cross prediction, which is also a key achievement that we are able to do in the yam breeding. The cross prediction, what we use here is kind of like a prediction model, but at the same time, we use it to estimate the crossing merit of 2 parents, what we use to advance our progeny from one generation to another one. So before in yam, before we could start making crossing, we have to wait at least for 4 years. But with this method, we are able now to minimize the crossing cycle from 4 to 3. This is the model we use. And also we estimated the crossing merit among the parents, and then which vary from -2 to +4. And then we base our selection from any cross combination between 2 parents, those cross that display crossing merit above 3. So we make our selection, which was 188. Then we added some parents and then make a crossing design. And then we planted them this year, and then we observe a very good performance, especially on the hybrid vigor, which means this method also can be really used in terms of plant advancement and then also selection. And then you can see using that method, comparing the one we selected and then the one we did not select, you can see that the material we selected that display a very high performance compared to the one we don't select, which means we are also making a very good progress. So as a lesson learning or the taking home message, what we are going to share here, just several sequence platform has been used in the yam breeding program, and then we documented everything. And then the SNPs call a roadmap been also been implemented, and then we know, okay, for this one, this is the way to go about it. And then genomic method, we have fully implemented then in parent selection and then advancement. And then the key SNP region that are controlling some of our traits, we know them, and then we are using them also in the breeding program. Quality control marker is available. We have right now 50 SNPs marker that we can use for routine activity in the breeding program. All these ones are already available on EIB website, so you can use it. Marker assisted selection implemented in the breeding program, and then genomic prediction also is also implemented. So thanks so much. So for this one, we are really grateful to DMGF and also to some Intertech, EIB, and also to the Polyploids group also. Thanks so much.